who's here to talk about Varroa and how it develops in bees. I run through a long piece of work looking after and seeking Varroa resistance through breeding and support and management of bees. Rhona manages about 60 colonies in South Worcestershire and has come down especially for this, so thank you. Staying with Helen. Her bees haven't been treated with Varroa since 2007, and I've been part of it, again, a proper scientific research study, Professor Stephen Martin's research into Varroa resistance. A member of the Varroa resistant UK team, a member of the Bee Farmers Association, and a BBKA master beekeeper. Rhona, thank you, we're really looking forward to the call. Please store up your Q&A for afterwards, this should be a good one. Thank you.
Thank you very much, and thank you for the invite to come and have a chat with you. It's a February evening, a wet February evening, so it's a lovely sight to see so many of you here. It's a subject that for some of you might be controversial. Some of you may already be doing something similar to what I've been doing. But this evening I'm going to sort of chat to you about, I suppose, why I went down this route and what we did, and give you a few tips on what you could do should you wish to follow that route, and also tell you about some of the pitfalls and some of the things that you probably shouldn't do if you want to keep bees. So let's see. So we're going to talk about our approach to Varroa, and we'll start with a little bit about me, I suppose, and my bees. So it might come as a surprise, but I'm keeping bees in Worcestershire. I know the accent doesn't quite match. I've never actually kept any bees in Scotland, although my beekeeping experience is from Worcestershire. Started beekeeping in 2000, very conventional start, local beekeeping association, beginner's course, and just followed what I was taught. So got local bees, got national hive, which is what most folk in our association keep, and sort of went down that typical route most folk go down. Where I was working at the time, there were free living bees, and it's strange that when you become a beekeeper you start to notice different things. I had already been there a few years and I was completely unaware of these free living bees, but they're a significant influence, and I'll talk a bit more about them in a moment or two. As you can see there, from 2007 our path went along a different route to what we had been taught, and it's grown from one colony initially to a small scale commercial enterprise. So anyone who's a new beekeeper here, beware, hobbies can get out of hand, okay? I'm in Worcestershire, and that's typical Worcestershire countryside. Quite a few of our apiaries are in orchards. We're quite a rural county. Devon, this sort of area is very rural, but it's, from what I've seen, I haven't seen very much of it, I've only seen a bit today. We're a little bit different. We're quite a horticultural area. We're right in the centre of the country, so we tend to get extremes of weather. I'm based around Pershore, and you'll sometimes see Pershore on the national weather forecast for extremes. So we get the coldest in the country, so we get -19, we get the hottest, it's 40 degrees, we get drought. So we have, I suppose, quite a different climate to what you guys have down here, but we're still very rural, and all of our bees are in a rural agricultural and horticultural landscape. And I can't fail to mention that's Richard, who's hiding in the back corner there, and he's very much part of it. So we beekeep together, and started out on the beginner's beekeeping course together. Okay, so why did I veer off that path in sort of 2006, 2007? Well, as I said, I started to notice feral colonies living in buildings where I was working. and they fascinated me. And when our employer found out that we were beekeepers, well, it was automatic that when these colonies swarmed, we would be called to come and get the swarms. So a lot of our bees started to be swarms from these free-living colonies. I also had the opportunity to hear a talk by Ron Hoskins. I don't know if any of you have heard of Ron. Unfortunately, Ron's no longer with us. An amazing chap. Total dedication to his bees. He died at 92, I think, a couple of years ago. And even up to a fortnight before he died, he was in his apiary or his bee shed every day, you know. And his focus was Varroa resistance. And a lot of what Ron was a pioneer in looking at has come true. Not all of it. There are a few sort of red herrings along the way, and I'll come back to those. But I saw a talk by Ron and what he was doing, and some of the pennies started to drop, and I sort of thought, well, if Ron can do it, and if I have offspring from these free-living colonies that were surviving without any Varroa, then perhaps, you know, I could get to where I wanted to be. It was also a time where there were changes happening in the Varroa that were on the market, and a lot of the traditional pyrethroid Varroa were starting to fail, and that's for a different sort of resistance. So the Varroa mites were becoming resistant to those chemicals. So there was lots of changes happening, and it just felt like it was not the right way to go. I come from a horticultural background, and it felt like we hadn't really learned from some of the experience of horticulture and agriculture, that you get onto a chemical treadmill and you can't get off of it. You know, when a chemical fails, you need to then look for the next one to sort of stay where you're at. So I wanted to go down a different route, and that's where the sustainability comes in. And that's one of the swarms from one of the feral colonies that we collected. So our bees, they're Worcestershire mongrels. They look very varied. So we haven't selected for colour. Some of our bees are very dark, some of our bees are very amber, and everything in between. We've got an open mated population. We've never done any artificial insemination. We're keeping local bees. They typically do well in a single national. Some of them are a bit more prolific. If they need more space, we give them more space. One thing they do have in common is they're all pretty good at propolis collection. So they are quite sticky bees, which not everyone likes, but I always felt that they were doing it for a reason, and the science is starting to sort of show us that now. I did have several beekeepers tell me that I needed to find better bees that didn't, you know, collect all this propolis, but I'm pretty glad that I ignored their advice. So let's see, how do we start? This is probably familiar to anyone who's been keeping bees. So this is a graph out of the NBU's Varroa management booklet. Yeah? They've changed the colours in the sort of latest one. This was the one that I had when I first started, but the graph hasn't changed. And if you think back, if anyone's been keeping bees for a long time, if you think back to sort of 2007, Varroa resistance was not anything that was talked about, and there weren't any books or websites or papers that you could go to for advice. So the only information that we had on Varroa was what was published by the National Bee Unit. So we took their booklet, we learned as much as we could from it, and went from there. We have made mistakes along the way because we didn't, you know, have anyone that we could go and ask, if you like. So we used this, and this graph sort of guided us on, you know, where colonies are in terms of managing Varroa. So if you're having a mite drop that's up in this sort of red area, your colony is in trouble. Okay, we would agree with the NBU on that. if your colony's down in this green zone, then they're doing brilliantly. And then you've got a sort of amber zone in here where they might be okay or they might not. You need to sort of watch them carefully. So we were trying to keep our colonies out of this red zone. And our monitoring, we would monitor and sort of record and manage anything that was up in this red zone and try and keep everything down green or amber. So the advice at that time was to monitor 3 or 4 times a year. That's still the advice. I checked on the booklet today, it's still monitor 3 or 4 times a year. If you're going to start looking for Varroa resistant bees, that's not enough. The more monitoring you can do, the better. 3 or 4 times a year, you will actually miss some key data points, if you like, and the more data you've got, the more informed plan you can have. So 3 or 4 times is just not enough, I would say. What did we do? Well, we did the monitoring, and what did we see? We saw big differences. And when the NVUs say you can't just monitor a single colony in your apiary, you need to monitor them all, they are perfectly right about that. Some colonies can have huge mite numbers, others very, very low. So you do need to monitor all of them. One odd thing that we saw, and we really didn't know what to make of it, was lots of immature mites on the board. So we were putting in a monitoring board into our open mesh floor and doing the monitoring that way. When you're counting mites, you need to only count the dark red ones. So if you get any that are very pale, so sort of off-white, sort of creamy colour, you don't count them. The booklet never said anything about immature mites, and we didn't know that there was any significance behind that, but it is actually quite significant. But if you're counting your mites, only count the red ones. If you count the immature ones, your figures are going to be very out. Our initial approach, so what did we do? So we did the monitoring, we continued to collect swarms, and we prioritised swarms from feral colonies. So we knew of some that we were always very keen to get the swarms from. There was another beekeeper that also worked at the same place, and there was a bit of competition to get those swarms, and it was, you know, who could get to it first. But yeah, we've actually both ended up going down the same path because of the bees. We've both had the same bees, so we continued to collect swarms. Something that was very common at that time that you don't hear really anything about these days is the use of icing sugar. So we dusted icing sugar onto colonies during mainly the spring and early summer, and the idea behind it was that the bees would groom the icing sugar off themselves and knock mites off. It's something that you certainly don't do late summer if there are wasps around, because it's only going to attract wasps to your apiary. So not really sure why you don't hear anything about it. It's really only a very minor means of control. It's not going to, you know, replace a varroicide, but we did it on a regular basis when we sort of first started. If we had a colony that was up in that red zone and had high mites, we would step in, but because we'd stopped using varroicides, we used biotechnical control, and shook swarms are as effective as varroicides. So if you want to give up varroicides, but you're still going to manage your mites, you could use shook swarms. You need to have a strong colony. The weather needs to be good, and there either needs to be a nectar flow or they need to be well fed. But it's very effective, and it's great if you need to change a comb, you know, so you can achieve multiple things. But if you've got a small colony or it's the wrong time of year, it's not going to be suitable. And those colonies that kept the mites low, we raised queens and drones from. So we tried to flood our local area with drones from these good colonies. Every colony that we had had a frame of drone foundation, or they had a shallow frame that they could build drone comb below. If they had high mites, we would cut that out and dispose of it so that we didn't contribute those drones into the local population. If they had low mites, then those drones were allowed to go and hopefully pass on their genes. So those two methods are biotechnical control. You don't have to use that at all. There are, other ways around it, but that's what we did. What did we notice? Well, we noticed that the mite drop decreased over the years. As I said before, the immature mites, we saw those, and we started to, many years later we found out the reason, why we were seeing them. I'll come to that. We would occasionally see bees flying out the entrance with a piece of bee pupa. We didn't understand what was going on, but we always sort of felt it was something right and good. Couldn't explain why, but we sort of felt, they're doing something that's good. So that is indeed true. Our winter loss is typical for the area. So when our local beekeepers had a bad year, we had a bad year. When they had a good year, we had a good year. So we were in tune with what the other members of the association were experiencing. Something else that we noticed, and again couldn't explain, was bald brood. So back in 2007, if I mentioned the word bald brood, you would automatically think that it was either caused by wax moth, or it was a genetic condition that you should requeen for, was the standard advice. That's advice that I ignored, because we were starting to see a lot of it, and again we couldn't explain it. But this bald brood is actually really quite important and is at the heart of what we now recognise as resistant behaviours. So this is bald brood that's associated with wax moth. You can see that line there? Yeah, so you can see the sort of silk path that's left. So the cappings have been removed, but the silk of the pupa is left behind. And when you uncover it, you know how to get the wax moth out, the larvae out, by tapping the side of the frame and out they pop. So that's the culprit there. So that's one type of bald brood. But this is bald brood associated with Varroa resistance. It looks a little bit different, doesn't it? What's different about it?
Not a straight line.
You haven't got a straight line. What else haven't you got?
They've got ridges around the south.
Yeah, so you've got little sort of wax ridges round these cells. You haven't got a line.
No silk.
And you've got no silk. So this is bald brood associated with Varroa resistance. You can see.
You uncatch those with the dished middles to the cells.
I haven't done anything. The bees have done that.
Okay, because Steve Riley reckons it when we tried it as well. If you uncap those with the dished centres to the cells and look underneath, those are cells without any silk lining. So they've actually been uncapped and recapped, but the silk lining has been removed.
Yeah, I've got a better photo in a minute, but here, I'm going to tell you a little bit about recapping in a minute, but you can also notice that these cells here are not your classic domed brood cell. When you open a textbook, a beekeeping textbook, and you've got a photo of healthy brood, it's wall-to-wall brood and it's domed cappings. That's not what we see in Varroa-resistant colonies. So Varroa-resistant colonies, the brood starts off being domed, but if you start to look at all these, you start to see that they're sort of dimpled. and they're flattened. And if you look carefully, you can sort of see different colours of wax on these. I've got another photo I'll show you in a minute, but that does not look like a photo out of a beekeeping textbook of healthy sealed brood. Okay. And you'll also notice that we've got some missing cells. So these missing cells, a lot of them, it'll become evident in a minute what's happened to them. So there's a comparison the two. With wax moth, you can sometimes get these sort of cells with the rim around them, but you usually have that line of silk. And with the bald brood, you've got more of a random pattern or individual cells, and you can see the pupa looking out at you. Right, the bald brood then, you don't see it on every frame, so the amount varies throughout the year and the amount varies colony to colony. It's perfectly possible to pick a frame out of a resistant colony and it look like your textbook frame. You could go to the next frame of the colony and it will look nothing like the textbook frame. But also throughout the year, the amount of this activity does vary. So, I'm going to do a little bit of biology, a little bit of revision. You'll all know this, won't you? Yeah, nod your heads. Okay, so top left up here, that's a Varroa egg. That's a Varroa male. These are two immature Varroa, and that's your adult female. That's the one that you're counting if you're monitoring Varroa. Let's see if, of course you're monitoring, aren't you? So that's your mature female that is going to lay the eggs. We've got a little sequence here that I have borrowed from the NBU booklet. Basically sort of showing you what the Varroa mite does. So that mature female goes in and hides in the brood food and waits for that cell to be capped. So that's your bee larvae there. Once it's capped, it's about 70 hours from her entering till she lays her first egg, and that first egg produces the male. After that, about every 30 hours, she will lay an egg and they will be females. And she can lay potentially quite a few eggs, but the thing that's restricting their biology is actually the time frame that the bee is in sealed brood. So really, Varroa mites are not particularly well adapted to the western honeybee. They're non-native, as you know, so they're adapted more to their native host, Apis cerana, but in the western honeybee, she lays a lot of eggs that will turn into immature females and will never actually emerge. Typically, in a Varroa susceptible colony, you only get 1.7 successful offspring, female offspring. The males never leave, the males die in here after they have mated. So it's 1.7 female offspring. If they're reproducing in drone brood, you get 2.2. These are averages, and there's a lot of variation within biology, so not every female mite that goes in will be successful. Sometimes they don't produce a male, sometimes they don't produce any eggs, sometimes they're so slow in producing any offspring that it doesn't work, so that's why there's sort of these odd numbers here. And it takes 5 to 6 days from an egg to an adult male and 6 to 7 to produce a female. So you can see that after the sort of 12 days where she's got an opportunity to reproduce, there's not actually, very much time to actually achieve it. So if we go on and we look at what happens in a resistant colony, we've got some specialist bees that do things that bees in non-resistant colonies don't do. And the first of those bees are detector bees. These are bees that can come along and they can have a sniff it's not quite the right way to put it because obviously they're not sniffing with their nose, but you know what I mean. They're detecting volatile compounds that are coming through the wax cap that are being emitted somewhere in here. There's a little bit of debate where the volatile compounds are coming from, whether they're coming from the Varroa offspring or whether they're coming from the bee that's been damaged by the Varroa offspring, but you only get it when you've got Varroa offspring. So if there's a mother mite goes in and she doesn't produce offspring, these chemicals are not actually produced. So that's quite an important fact. So this specialist bee comes along and she detects that there's a problem here. She doesn't quite know where the volatile compounds are coming from, so she starts to open these cells to get a better idea. And if the remover bees then come along and they don't have quite such a good sense of smell, but the cap is now open, they know that there's a problem in there, they will actually deal with that pupa. And it's a little bit gruesome. It's difficult for them to get the pupa out whole, so it's chewed down. So this goes back to what I was saying about seeing bits of pupa being flown out of the entrance of the hive. We start to sort of fit bits of the jigsaw puzzle together. But that's how they get it out. Now at this point, the foundress female that went in and has laid some eggs, etc., she has not had enough time in there to produce mature female offspring, because this is happening about 5 to 8 days after the brood is sealed. At that point, there are no mature female mites. So she escapes. She's not killed, the bees can't detect her. She gets out as that bee is being chewed down, and she still has an opportunity to go and do it again. But these foundress mites typically only have 2 or 3 reproductive cycles. She has used one of these reproductive cycles here, so she's gone ahead and she's laid a load of eggs in here, but been unsuccessful. If she goes in, let's say, to the next cell here and does the same again, she might get picked up again, and she might have burnt her bridges and used all of her eggs by that point. So one thing that we actually find in resistant colonies are a lot of infertile mites. Now it's not something that you can, pick up a mite and go, yeah, that's fertile or infertile. It's something that has been done by the researchers, but these are mites that basically had attempts at reproducing, been disturbed for whatever reason, and are not dead. They're still running around the colony, but they've not got any eggs, they're not able to reproduce. And in resistant colonies, you've typically got 3 times the amount of infertile female mites you'd have in a susceptible colony. And this kind of explains why. Now the bees don't always get it right, so you have a third set of bees called recapper bees that come along, see that the cell's open and go, what's going on here? Let's cover this back over, this shouldn't be open. Okay? They don't have the sensory abilities of the detector bees or the remover bees. They've just got the job of covering over and, well, this is open, this shouldn't be open, let's cover it over. And you want that to happen to the mistakenly open ones, but you don't generally want it to happen to the one where the pupa should be removed. However, we're back to the beginning, and the detector bees are still on patrol, and she could come along and detect that a second time. There's some research from Germany that shows that there are some colonies that do multiple opening, and there's one, a record of a cell being opened 17 times. Now it's not an ideal scenario. You want them to get it first time. So it's imperfect. And the fact that we have 3 different types of bee involved in this activity also explains some of the difficulties in getting Varroa resistant bees, because some colonies can do some of these activities and not all of them. And if they only do some of them, you haven't got a resistant colony. So you can have colonies where they detect there's a problem and they come along and they recap it. If they don't actually do this stage, then they're not actually resistant. And you can look and you can see the bald brood and you go, yes! But if you're not getting that activity, they're only part of the way there. So you need to be careful what you're looking for. So a little bit of photographic evidence. So you've seen these bees, these pupa looking out at you. Typically, most folks see them at this pink or purple eye stage. Some of our colonies do it earlier and the bees have white eyes, so the colour develops with time. It's actually really useful to do it at an early stage, but I won't go into that at the moment. Typically it's pink and purple eye. Here you can see these cappings that have been replaced. So you see these dimples and the slight changes in colour of the wax. That's where the recapper bees come along and replace the wax. And hopefully only done it on the ones that don't have Varroa. Here on the right hand side, what have we got going on here?
There's a partial. We chewed out.
So you've got a chewed out pupa. You've got half a bee, alongside ones that are sort of looking out at you, and alongside ones where there probably was a pupa in there and it has been removed. So that is key, that chewing out. And I have actually pulled that out. The bee hasn't pulled that out. I've pulled it out to show you what is actually going on in there. It's pretty gruesome, they're sort of headless. But one thing to mention is that it looks very different to typical brood disease. So most brood diseases we're looking, if it's EFB, we're looking at larvae. Here we're looking at pupa. So in every respect that looks healthy, apart from being dead and headless. So other signs of Varroa resistance. This is a photo from Steve Riley, who has written a very good book, and Steve has looked very carefully at his Varroa inserts to find bits of bee pupa. These are really difficult to see if you have white insert trays. So you can only really see it if you've got a dark tray, or you take the debris and look at it on a different surface. But a dark insert tray can make it difficult to see Varroa. So it's a bit of a compromise. It kind of depends how good your eyesight is. So signs of Varroa resistance. The only one that you actually really need to look for, you can ignore what I've just been telling you, is look for a low mite population. So you don't need to go in and look for bald brood or cannibalised brood, because you could find that you've got a colony that's only got some of the traits. But if you have a colony that has a low mite population and is maintaining that low mite population fairly consistently with a few ups and downs, then you're on the right track. Bald brood and cannibalised worker pupa, pepper pot, brood pattern, pupil parts, and immature Varroa are nice to see, but you don't have to go looking for them. So that's going back to your monitoring.
Sorry, on that picture, are there any immature Varroa? Couldn't see any.
I can't see
No.
any, no.
Sorry, I was just checking I wasn't missing it.
You've got a bit of antennae, these antennae where they've sort of bitten them off or sort of pulled them off. These are what Ron Hoskins was looking for. So a lot of Ron's work was looking for colonies that produced what he called these hockey sticks. and yeah, there's he spent a lot of time counting bee antennae.
Do they consume the pupa that they take out? Do they actually? Is that quite a lot of protein that they're losing?
Yeah, I mean, bees will cannibalise drone larvae, and if they're short of protein, you know, if drones are not required at certain times of the year. Don't know about this. I haven't seen anything that said either way. It might depend. It might be that they do it sometimes if they are a bit on the hungry side, but other times they're just going to dispose of it. A common question is grooming behaviour part of Varroa resistance? This was another thing that Ron was convinced about. And you sometimes see photos of Varroa with dimples on them, or with sort of splits and missing limbs and things like that. If it's dimples, then it's actually a birth defect, and it's not related to the bees trying to get rid of them. If it's missing limbs or bite marks, for want of a better way of putting it, then it might be, but at the moment it's thought not to be a significant factor in resistance. It might be a minor feature. The other thing about resistance is that different populations around the world do slightly different things, and it might be that in some of the populations grooming is more important than in others.
Sorry, Rhona, can you go back to the previous slide? That regular dorsal dimples, developmental faults aligned with muscles, what does that mean? That's if it's a natural phenomenon.
That's a natural phenomenon.
Not caused by Varroa.
It nothing to do with the bees. So these dimples here on the Varroa, initially a lot of researchers thought that that was a sign that the bees were attacking the Varroa.
Well, certainly did, didn't they? Yeah.
It's not.
It's not.
That has been shown to be a birth defect. There are actually muscles under there that don't develop normally and cause that sort of pulling in.
Okay.
But if there were sort of missing limbs or chunks out of them, it might be down to the bees.
And is that QR code there, does that link through to the paper?
Yeah, there's a paper there that talks about regular dorsal dimples.
I'd like that please afterwards.
I can send you that. Selecting for Varroa resistant bees. Well, the pros, you don't have to buy Varroicides, you don't have the multiple applications, you don't have Varroicide risks either to yourself or to your bees. If you look at the sort of PPE you need to apply oxalic acids, vaporization and things like that, I look at it and I think, no thanks. So you can avoid that. Something else that might affect some folk is you might have to take your summer honey off to get a Varroa treatment on. If you go down this path, then that's not something that you need to think about. It's also fascinating and addictive, so be warned. The cons, you do need to monitor. Some folk with the best will in the world go down this path and decide they're just going to stop treating and everything's going to be fine. And unless you are really lucky and you've got good genetics from, say, someone else that's doing it, or from some long-lived feral colonies, the chances are your bees are going to die. Okay? So you do need that data to know whether you're in with a chance, and if your bees are not coping, then you do actually need to intervene. You need to manage the mites. It could be biotechnical, it could be Varroicides. If they're not coping, you might also want to give them away. So I know some folk that are very generous with the colonies that aren't coping and give them to a new home. But the other way to do it is that you requeen them once you've actually got the mites down to a suitable level.
How long would that process take?
The process from...
Well, if you don't treat, you carry on irrespective of that, how long would the colony take
To die?
Yeah.
It's a very good question. Some sources, the NBU typically say 2 to 3 years, but actually they can die a lot quicker than that. You can lose them in a year. You know, if you've got a colony that is doing really well, producing lots of brood, has none of these resistant traits, it will just be multiplying Varroa, and if you don't step in, you could lose them that following winter. So it can happen really quickly. Some colonies kind of hang on and they get through the first winter and you think, oh, it's fine. And then the second year, they just plummet and they don't make it through the final year.
So I haven't treated for 3 years now. Does that mean I'm on the way?
Are you monitoring?
Yes.
And are your mite levels reasonably low?
Yes and no.
Okay, so you've got multiple colonies
Yes.
and you're finding differences.
Yeah.
Okay. Let's have a chat at the end. You might not be out of the woods yet, but generally we say once you've got through 3 winters, you are heading in the right direction. It doesn't mean that you can't go pear-shaped at that point, but 3 winters is a good test.
Well, I'm coming on to the 4th year now.
Good. I, as part of my work with Stephen Martin, we were comparing my bees with bees that were naive for Varroa. So we had bees brought in from the Isle of Colonsay, which is a small island off the west coast of Scotland. They don't have Varroa, so their bees have never been exposed to Varroa. And it's a black bee haven, so you're not allowed to bring bees in, so it doesn't mean that it won't happen because other folk have ignored advice before, but generally, it's a island where there's no bees brought in. Bees do come out, they get sold for research because they are Varroa free. So we had bees from Colonsay brought to us to compare to our bees for these traits, and we got them in the September. We set the trial up. By the following autumn, the colonies that were headed by Colonsay queens at that point were just mite infested, and I naively thought I'll be able to save them because they were lovely bees. They were gentle, they were productive, they were lovely to handle. At the end of that trial, when we took the sort of final Varroa results from them and we looked at the sort of mite numbers, Professor Stephen Martin said, look, you've got to kill those colonies. And I went, no, we'll be fine, we'll manage them, so we decided that at the end of the trial, everything would be given an oxalic acid treatment because the numbers were just, up here. And I sort of thought, we'll select from the best next year and we'll sort it out. None of them made it through that winter. So that was basically, 12 to 18 months at the most after they'd had Varroa. That's how quickly a colony can die, and it was horrendous. It was heartbreaking to see, beautiful colonies collapse. So yeah, it can happen really quickly, but not they don't all die that quickly. So right, let's go on to something. What have we learned? We've learned that every colony is different, so there are degrees of resistance. Some do it better than others. Resistant bees are not invincible, so if you've got colonies that are coping and you have a colony in your apiary that isn't coping and you don't do something about it, that colony is actually a threat to the colonies that are managing. because the mites in that colony can go into your resistant colonies, and because some colonies are so good at dealing with mites, they go into that activity of detecting and removing infested brood, and they really go at it. And if they go at it hard enough, there's then not enough bees to get them through that next winter. So if you've got colonies in apiary where you've got mixed results, and some of them are doing really well and some of them are really not doing well, you have to manage those colonies with high mite numbers, because they're a threat to your resistant ones. So resistant doesn't mean invincible. Raising drones is important. You want to spread those good genes around, so raise drones from the colonies that have low mites. A lot of folk talk about brood breaks. Brood breaks are helpful, but they're not the answer. Okay, they're a bonus. So don't interfere in a colony that's having a brood break to suddenly, give it brood. That's not going to help, but it's certainly not your answer to control. And share with others your experience, your bees, your knowledge, and then it becomes easier for everyone.
Rhona, when you say raise drones, how do you raise drones? Other than not raising drones from colonies that aren't showing good traits, you presumably you would cull as much drone where you think they haven't got the traits.
So you could do as we did, give everyone a drone frame and cull the ones with high mites and allow the ones with the low mites to, carry on and live.
So just encourage extra drone from there.
Yeah. There's a photo here. If you actually look, these bees, I know they don't have teeth, but they've got their teeth into that pupa that they are removing there. So you don't see this very often, but they are, getting stuck in.
Do they manage to get it all out?
Yeah, it's an empty cell, and the queen will come along and relay. So our beekeeping is very conventional, which might come as a surprise. The few things that we do differently, perhaps, is that we don't do any routine queen replacement. So we do have old queens, and the consequences of old queens is that not every colony will make it. So some of them get too old and they just, haven't got enough to get through. But I like to have long-lived queens, and the only way you know they're long-lived is if you let them live. If you replace them every 2 years, you don't know how long she was going to live. I don't do any equalising of brood or uniting, and the reason for that is I want to know which colonies are doing really well and which aren't. So if I have a weak colony at the end of the summer, is it weak because it's full of Varroa or full of virus? And if I unite that with a small one, another small one that's actually doing fine, I could end up losing both of them. So I don't do that. And I suffer the consequences. Some colonies don't make it because they're too small. And I mark and clip my queens because I want to know who's who, and I also don't want my good queens to escape. And by clipping them, there's a chance that she'll end up under the floor and not lost if we fall behind on swarm management. I've mentioned Stephen Martin, that's Stephen there, and a couple of his postgraduate students that we were working with.
They look like primary school students.
He is particularly tall, but they're also both quite small, so yeah, they did look very young. I was assured that they were postgraduate students and not primary school, but yeah, I know what you mean. So these days we have sort of moved on from the monitoring sort of thing, and we're looking for other traits. A colony that comes out of the winter at its first inspection and looks like that, I'm very pleased with. So that was the 30th of March, I think that was two years ago. That's a big thumbs up for me, okay? When we have drone brood, you know, sort of under your queen excluder and you accidentally sort of break it open, we're always looking in the drone brood to sort of see, is it full of Varroa? It's not always a bad thing, because the Varroa that ends up in the drone brood is not in the worker brood. So as soon as you start getting drone brood, the Varroa mites head towards the drone brood and leave the worker alone. But if this was full of Varroa, I'd be a little bit concerned. So we're always looking, and then we're sort of glancing at frames, and you can sort of see the bald brood here in the middle of that frame. You can't have too much bald brood. And if you've got a frame where, you know, there's virtually all of it, you know, uncapped, you start to think there's something wrong. So you can have warning signs as well. So you're looking for subtle things as we go sort of further through. Right, nearly at the end here. So, am I doing this alone? No is the answer. And there are actually quite a lot of beekeepers who have followed this same path, and a lot of them have kept their head down. And they've kept their head down because this, which still surprises me, is a controversial subject. How we all want the same thing. We all want colonies that are healthy with low Varroa populations. There are just different ways of achieving it. But why that's controversial, I'm really not sure. So, the largest known group in the UK are up in North Wales, and there's about 100 beekeepers with 500 colonies that haven't been treated in the same sort of time frame as me. Okay, so they've been going a long time. And the way that their approach is really nice, in that all of their new beekeepers are given a nuke of local bees. So they stop bringing in genetics from out with the area, and they encourage the keeping of local bees. So that's been really successful. Western beekeepers down in Kent is Steve Riley, who's written a very good book, and I've got a copy here if anyone wants to have a look. Steve's very active and has supported Western and other sort of beekeepers in setting up apiaries to share useful genetics. Lune Valley in Lancashire. The Swindon Honeybee Conservation Group was what Ron Hoskins started. I'm really not sure what's happened since he has died. I haven't really heard an awful lot from
He's lost
them.
all of the bees. He lost the bees before he died. So there's no bee continuing.
Well, he lost some of them because there was a fire in the apiary,
Yes,
vandalism.
broke it, but he's lost all of his bees.
But there were other
He's
beekeepers
died
that
in
were.
the
His genetics are in the local area. They know that they are in surrounding apiaries. So, but worldwide, we're actually in some ways a little bit behind the times. In Cuba, there are 220,000 colonies that are Varroa resistant. So Cuba, poor country, couldn't afford Varroicides. They had no option. So they took the hit when Varroa came to Cuba. They lost a lot of colonies. They lost all their feral colonies, or virtually all of them. But the small number that survived reproduced, and the beekeepers collected swarms from them, and now they're up to 220,000 colonies that produce huge honey yields because they didn't intervene. There are populations in various countries across Europe. Here, this is Apimondia. So Apimondia was in Copenhagen this year, and I was lucky enough to go to Apimondia. This is the round table discussion. I know it's not a round table, but this is the round table discussion on Varroa resistance. They had security guards removing people from the hall because there were so many folk wanted to be in there. There were not enough seats, and they wouldn't allow folk to sit on the stairs or stand at the back. The place was absolutely jam-packed, and these are researchers from across, I think they're all European. That's Professor Stephen Martin, that's Steve Riley. They took a panel discussion, The guy on the end here, he's a bee farmer. He's a bee farmer from Austria. So the rest of these guys are research scientists. This guy on the end, after a suggestion by his wife, has gone down the Varroa resistant route. He didn't really want to, but he decided that for a bit of marital harmony he would go that way. He has 10,000 colonies in Austria. 2,000 of the colonies are in his Varroa resistant programme, where he is selecting for those traits. He monitors his colonies, his 2,000 colonies that are in the programme, 3 times a year. I'm telling you, you need to do it more. He only does it 3 times. He does the sugar roll monitoring rather than the natural light drop because it's faster. And he has a 3% Varroa limit. Anything above that is not in the programme, it's out of the programme. And he has been very successful. He's very happy he's gone down that route. So there are some very big players in it. Anyone who knows Randy Oliver in the States, a lot of colonies, and he's gone down that route. The Americans have done it differently. Like many things, they've done it differently to other people, and there are different models across the world as to how this has been achieved, but we know that there are an awful lot of similarities and that these populations are popping up all over the place. This is a new programme. The Germans have launched, this is the translation, but it's Varroa Resistance 33, with the idea that by 2033, all of the bees in Germany will be Varroa resistant. Okay? And when the Germans put their mind to it, you know, they're serious and they are bringing on board other European countries. They have a very different setup to us. They have breeding stations and, you know, a lot of bee research involved, and I reckon that they will achieve it. They are very serious about it. Here in the UK, beekeeping education has started to embrace it. So these are a group of NDB students, National Diploma in Beekeeping students, looking at a Varroa mite drop from one of my colonies. BIBA have launched Varroa resistance through bee improvement, so they've got a lot of support for members, and the BBKA have integrated it into 3 different modules for anyone who's doing modules. They've sort of taken that on board, so it's definitely becoming more mainstream. Finally, suggestions for anyone who wants to go down this route: monitor. Don't do it without monitoring. You'll have a terrible experience if you don't know whether your colonies are coping. And keep records. Don't think it was colony number 1 that was doing well, wasn't it? I'm sure, or was it 2? Okay, you need to know. Ideally, you want your mite fall somewhere low at the end of the year as it was at the beginning of the year. Steve Riley reckons about 5, an average mite fall of 5 per day is what he sort of aims at, and that's a pretty good measure. There will be peaks and there will be sort of troughs around swarming time, but as long as it returns to sort of normal levels and doesn't just go up, then you're on the right path. As I've said before, raise queens and drones from the ones that are actually doing it right, and don't raise them from the ones that haven't. And if you find that your bees are not showing these traits, they're not coping, then your only sort of other options are to work with others who are perhaps having more success, or to look for genetics from long-lived, free-living colonies. And if you take photos, the wonderful invention of the camera phone, you know, I know you get propolis on it when you've got it in the apiary, but take photos, go home and enlarge them, whether it's a photo of your mite drop board or a photo of a frame with bald brood on it. You'll see so much, and it will start to sort of make more sense. You'll see things that you've just never noticed before. Photos make it fascinating. Oops. There are lots of resources out there. I've got a couple of books with me. You don't actually have to buy anything. There is a Varroa Resistant UK website that's got advice, it's got scientific papers, it's got an email address, so if you've got questions after today and you want to fire it to the team, Professor Stephen Martin is the sort of head of the team, then we will do our best to answer them. That's Steve's book. This is a new book that's out, which I haven't managed to read all of it, but what I've read is pretty good. It is a book written by a Polish beekeeper that's been translated, but there's a lot of useful information in it. So do have a look at those if you are interested. And otherwise, thanks for listening.
Thank you.
Rhona,
Yep.
with your experience, is it true that these bees don't travel well? Because our problem is where do we start? Where are those colonies? You were fortunate in some respects in that you actually got swarms from feral colonies, so that gave you a head start.
Yeah.
Now, as you know, I and Rhona have been working on this for the last, I don't know, 5 to 10 years. Not making that much progress. I had 2 colonies 2 years ago that were definite uncappers, recappers, anatomical things on the floor, and I lost them that winter. I didn't treat.
Were you monitoring the mite?
Yeah,
Yeah. And mite numbers were low.
yeah, but I lost them.
So bees die for lots of reasons.
Absolutely. And now this year, I don't know if it's the same way you are. I went into the winter with 12 colonies. I'm now down to 2. I've never ever in my, whatever it is, 36 years of beekeeping, lost that number of colonies.
I don't know whether this will make.
Across the association.
Well, I don't know whether this will make you feel any better, but there are significant losses in our area. So within our branch and our neighbouring branch, there are significant losses, and there are beekeepers like you who have had massive losses. Some of them, I had a beekeeper on the phone the other day who's been keeping bees for 50 years, and he's lost all of his colonies.
Lost them all.
He's lost them all, and he was on the phone, could he get a nuke of bees from me? He said he's never been in this position
No, I
in
know.
50 years. So
But, you know,
it's
where
not
do you
looking
get-
good.
Where do you source those resistant bees from? Because I think what we ought to do as an association is now identify all of our feral colonies and surround those colonies, not remove them, but surround those colonies with bait hives and actually see if we can capture some feral swarms.
Long-lived feral colonies, so you need to know that they're long-lived, because feral colonies can be, you know, a beginner swarm that just appears and then, you know, dies out. If you've got colonies in houses, homeowners generally know if the bees are alive during the winter. So colonies in houses, making them a valuable resource, and like you say, putting bait boxes in people's gardens to collect swarms from them.
And what people tend to do is remove those feral colonies, but I think that's the worst thing you can do.
It's the worst thing you can do,
Leave
and
them alone,
you
and
need
they
to
capture any swarms from them.
treasure them.
Absolutely.
And leave them, and I try to tell homeowners, look, you're really lucky that you've got bees in your roof.
Yeah, sure.
You know, they're not always that keen, but there are so many folk that can live for 20 years with a colony in their roof and it never causes them any issues. The trouble usually comes when I'm about to sell the house. What if the
Yeah.
new owner doesn't?
Or I've got grandchildren, and they're coming this weekend, get rid of them for me.
Yeah. So that's one source. Research seems to show that there are resistant traits in the general population of bees at a relatively low level, but within an association, you guys, if you start looking, you
Yeah,
should
we
be able to
have
find.
them. We should have them.
So even if you haven't got these feral colonies, if you start screening your colonies, so the regular monitoring, the keeping records, the comparing, oh, I've got one that's managed to keep the mites down at 5 per day for the whole year. Raise some queens, you know, share the spare queen cells when they're swarming, don't squish them all, give them away or make some splits, or you know,
We've got to do that collectively, guys.
it's much easier the more folk that are involved. It's really quite tough doing it on your own, because there are ups and downs. And the more folk that are involved and the more folk that are actually selecting for the same thing, the more successful you'll be and the quicker you'll get to that success. If you can sort of treasure your local genetics and discourage folk from bringing bees into the area. Now this is going to be really tough if you've had a tough winter and folk are wanting to replace their losses. You can go onto the internet, you can, yes, buy a nuke of bees that's got a lovely Carniolan queen or a Italian or a Buckfast or a whatever. Those genetics are coming in and they are diluting your local genetics. That is slowing progress in that direction. If that's the direction that you want to go in, you need to support those that need bees by providing local bees in some sort of way, whether it's swarms, distributing swarms, or actively splitting, giving bees or selling bees to beginners, that sort of thing.
But my original question was, in your experience, do these bees travel well? Because I've had queens from Ron 15 years ago, 20 years later, I'm from Ron. They didn't perform well in my hands. We've got local beekeepers that have not treated for one guy for 20 years. I've had queens from him, I've taken grass from him, they haven't done very well in my hands. Where do you get that sort of resistant bees that are locally adapted that we can then breed from?
That's a really difficult question because there is quite a bit of evidence to show that when you move them to another area, they don't perform as well as they were performing in their original area. Whether that is down to a local environmental change or how the beekeeper is looking after them in some sort of subtle way, I don't know why, and I have had this conversation with Professor Stephen Martin, and I haven't got an answer for you, but I know that there is evidence that when they're moved, they don't seem to be so successful. And in that sort of way, I get asked quite regularly if folk want to buy bees from me. And I'm really reluctant to sell bees because I know that if they fail, they'll go, well, they were just rubbish. And I don't want to give folk evidence for it to fail, for it failing. So I would rather encourage folk to nurture what they have locally than try and buy in. There is one big bee farmer who will remain nameless who is seeing an opportunity to sell resistant bees, and he wants to get hold of resistant bees to then be able to sell them to the mass market. I'm not sure that that's the way to go, and I declined his offer of buying bees, and he has approached a number of folk to buy bees off, and we have basically said no.
The trouble is, there are so many beekeepers that will buy from the commercial bee farmers, and as you say, they're going to be constantly flooding. I'd rather they were flooding with your bees, even though they maybe don't travel so well, than the ones with imported queens. It's a difficult one, though, isn't it?
It's a really difficult one, but the fact that we have populations in different parts of the world, in different parts of the country, that have been successful shows that you can do it anywhere. And it's not one type of bee that does this. Different subspecies, etc., in different parts of the world have been shown to sort of do this. So we've achieved the same thing in sometimes in different ways in different parts of the world. So that's why I think you need to nurture what you've got locally as much as you can.
I agree. I think we've got to do an association-wide identification of feral colonies and then see what we can do.
Let's set something up for Beekeepers Day and what we need to do in order to be ready for Beekeepers Day.
I mean, does anybody here know of any feral colonies?
perhaps not because some of the Peter Taylor Church,
Okay.
they tend to be a bit on the aggressive side.
Well, that's all right.
When it comes to selecting, if you're going to go down the route of selecting for Varroa resistance, you might need to park some of your other selection criteria till you've got things stabilised. You know, if you have half a dozen selection criteria that you want them to be this particular colour and you can handle them without a smoker and you can X, Y, Z, you're never going to get there. So resistant bees can be as calm can be, and they can also be horrible, and everything in between. If you get a resistant colony and it's one that you just need to make sure that you are zipped up and you've got plenty of smoke, you can work on the temperament. You know, you can raise daughters and select the best daughters and go from there. It's something that can come.
What about the these queen breeders that are advertising hygienic behaviour and grooming and what have you? They're obviously not advertising specifically for mite resistance, are they? But
Good question.
they sort of are implying that, and people go, oh, I'm going to pay £95 for a Varroa, extra Varroa resistant queen, or very, very hygienic behaviour queens. And I think that's a bit of a con as well. To be honest,
So,
how can they prove that?
if you look on the market, you probably won't see anyone selling Varroa resistant queens in the UK, but you'll see hygienic, hygienic behaviour or some sort of similar term. There are relationships between hygienic behaviour and Varroa resistance, but hygienic behaviour is a broad suite of behaviours designed mainly to deal with sort of brood diseases. And in the States, they went down the route of, or a lot of them went down the route of, well, let's select for hygienic behaviour and that will take care of Varroa. Some of them have kind of got there in a roundabout way, but a lot of the hygienic behaviour, they are killing brood, either by sticking pins in it or freezing the brood, and then you go back a certain number of hours later and you look at how many pupa have been removed. What you're doing is you're selecting for bees that can detect dead brood. The Varroa is not killing the brood.
No.
Those specific ketones and acetones that are given off in those Varroa infested cells are not the same smells from dead brood. So you're selecting what can be quite useful traits, but it's not the same thing. Some folks start with that, they find bees that have got hygienic behaviour and then they start selecting for Varroa resistance from them, but they're two different things, and it's very easy to look at the marketing, buy these fantastic hygienic behaviour. It's not the same.
If you buy Varroa queens, Varroa resistant queens, how long do you think it would take to actually get to the point where you can say you've got resistant colonies?
Within a few brood cycles, that colony is resistant. So the experiment that we did with the colony bees came and we swapped the queens from my colonies and the colonies over. So we ended up with colony bees with a Worcestershire queen, and they're the ones that survived. The ones that then went on and died were my bees with a colony queen. Basically, within a few generations, once you've got that first brood that has emerged from that resistant queen, those daughters will be doing that activity. They will be patrolling and removing anything that's a problem.
So why don't we do that here with getting resistant queens with our natural colonies? Would be great.
You've got to have the resistant queen.
Yeah, and it's also the inheritance. So we know that it's inherited down the queen line. The inheritance is complicated because bee genetics is complicated. Let's start there. But we also know that it's not just straightforward Mendelian inheritance of genes. We know that there's epigenetics involved, and that just it means that the genetics is complex and subtle at the same time, that these traits can be reinforced in a queen in her life, which kind of blows my mind. And those little tweaks to her genetics can then be inherited. You know, the epigenetics thing, I don't fully understand, and but I'm assured that that is part of how it's inherited, and it's part of the complication of raising queens. The other thing is you do want drones from suitable colonies, and if you are surrounded by drones from imported bees, that is going to dilute what you've got, and that slows things up and makes things much more tricky. Sorry, there's not a simple answer to it, but there is a lady here who's been trying to tell us.
Hi, thank you. My name is Ruth and I'm the Education Officer for Oakhampton Branch. Thank you for the invite, Scott. And I'm involved in teaching the beginners on the basic. And I wanted to ask about the Varroa calculators and what you think about the two, because they are quite different and beginners are quite a vulnerable group.
Yeah.
And the MBU basically are saying, please keep your Varroa numbers below 1,000 in your hive.
Yep.
And the Varroa resistant Steve Martin stuff is at certain times of the year, like midsummer, saying it's fine to have 12,000 in there because these bees are short-lived. That's quite a big difference. And I'm wondering how best to advise that vulnerable group of beekeepers. My instinct is to be quite conservative and say, please just treat your bees, learn how to be a beekeeper for 3 years and treat, and then after that maybe try. But I actually want to make sure that I've heard it from somebody that knows an awful lot more than me.
Okay, so the NBU model was originally designed by Professor Stephen Martin. So it's his data from 30 odd years ago that the NBU are still using. His data has been validated in recent years, but what the NBU have done is they have are you still filming?
Yes.
They have
Being conservative.
manipulated the interpretation of the data, let's put it like that. So he stands by his original data and the original model, which said that a population of 2,500 mites is fine, and above that you're in trouble, unless there are short periods in the summer where it can go above that. The NBU have sort of allowed the level to drop to 1,000, and they've said that anything above 1,000, you know, you're in trouble. So it's really tricky for beginners. There's a lot to take on board. I have a group of beginners who I have done a beginner's course and then I've mentored, and they had some bees from me, and so they've never treated. So it is possible to start right from the word go on this path. But if your beginners don't have either the genetics or the close mentoring, it can go wrong. So being conservative is fine. I think getting them into the swing of monitoring once a month or more if they can, would be a really good habit to get them into. And then if they notice that they have bees that aren't actually reaching that threshold, then it's a winner. So it shouldn't come as a new skill, the monitoring. If you can get the monitoring in from the word go, then they've got the skill to make at some point the decision whether they want to treat or not.
Thank you, because my basic thought is that they need to have bees in order to understand bees.
Yeah.
And if they die because they haven't treated or they've worked on numbers that are just a bit too in the risky zone for them, then they won't get those skills.
Yeah.
And it's game over very quickly.
Yeah.
Seems to me that one of the biggest takeaways is that if you want to go down this route, it's not a do nothing option. You know, you've still got to be absolutely active in managing Varroa and controlling your Varroa. You're just doing it differently.
Yeah, it's
And
not about leaving them alone. And
leaving it alone and walking away is not
that's
the option
not
in any of these.
what I'm
No, absolutely.
suggesting, you know.
No.
You're actually being more actively involved with your colony because you're really getting to know your colonies, and you will know that colony number 1's fantastic. Look at that low mite level. Colony number 3, she's for the chop. You know, you really get to sort of know them intimately, and for folk that have perhaps been beekeeping for 30 or 40 years, it can be a way of reinvigorating and re-engaging with your bees, because you think, oh, I've got the hang of this. Well, you know, have a closer look, get to know them really well, raise queens from those ones that are doing fantastic, manage the ones that aren't, and then requeen them. So, yeah, everyone can get involved, whether you're a complete beginner or you've been doing it for 50 years. It's not necessarily easy, but the more folk that do it and the more support that you have, the more likely you are to be successful.
And if you're monitoring and you use Varroa resistant, they've got an excellent calculator on there for what you should do at certain times of the year. If your mite level is high, obviously until you've got to the stage where you've got Varroa resistant bees, what would you suggest would be the best steps to take? What treatments, if any, should you be using if it's above the level that Varroa resistant suggests you can be at?
So that is your choice, really. If it's during an active season, then you've got more choices because you don't have to have Varroa. You could shook swarm. Your swarm control, if you do something like a pagden, then you're giving them a little bit of a brood break. That's going to sort of help. You could do ice and sugar. But at certain times of the year you haven't got those options, and with certain colonies you haven't got those options, and you then need to make the decision, well, which Varroa am I going to use? And the key time to use it is to protect your winter bees. You know, if your mites are only going in one direction, and you think, oh, I'll do the oxalic acid at Christmas, it might be too late by then. You know, if the mite's already high, you need to step in before then.
So would you do, well, I know you wouldn't, but would you suggest that we did oxalic acid in the autumn? Or should it be one of the awful strips?
Well, oxalic acid, you need them to not have sealed brood if it's going to be effective.
Yeah.
So you could create a brood break, you know, you could cage your queen. There are sort of different methods of doing it, either sort of the small sort of one thorn cell, or the full frame that you sort of retrieve the sealed frame and destroy it, you know, over a sort of month. So there are ways of creating a colony that is broodless or doesn't have sealed brood, and you could use oxalic acid at that point. I'm not the best person to ask about Varroa.
Fair enough.
You know, so I'm reluctant to give advice because I'm not speaking from experience. Sorry.
We're coming up to 9 o'clock, so I'm sure Rhona will take some more questions if people want to afterwards. If that's okay.
Yeah, that's fine. And I've got these books if you want to have a look at them.
But maybe others need to break away. Rhona, that's absolutely fascinating, and I think, you know, the engagement that we've got, and I feel the spark in the room to maybe start the project, which as Tony says, we've got to do with the critical mass if we want to do it. But maybe this first action, and I think let's as a committee we'll try and build something for the Beekeepers' Day to see how we do that. But one of the actions that I think we can all do that we've taken away from here is if we can all start finding and noting where those feral colonies are. That's a really good start. That will give, you know, we probably all, many of us will know that there's a feral colony out there, and it is the ones that have been there for a number of years. You know, because otherwise it's the swarm that I lost last year, you know, that it's just gone in the neighbour's chimney. You know, it's not those. Those aren't the ones we want. We want the ones that we know have been there for a few years because they've demonstrated the longevity and the persistence, the resistance, and not succumbed. But a hugely fantastic talk. Rhona, thank you very much.